thank you for the invitation to come and talk to you. I'm going to be telling you a little bit about our work on rapid virus detection and using single particle imaging methods and machine learning to detect and classify viruses. So here's the introduction to who we are. So we're in biophysics, and we're in the Clarendon laboratory, which is that nice picture of it before the Beecroft building was built in front of it. And what we do is we use a combination of kind of traditional biochemistry and virology techniques combined with single molecule biophysics methods to study viruses and to develop methods for their diagnosis and treatment. So before I get started, I actually have to say a really huge thank you to a team of people. So the story that I'm going to tell you tonight started around 3 or 4 years ago when we started looking at sort of very sensitive virus detection. And it goes all the way up to the present day where we're trying to use the assays that we've developed for coronavirus detection. So I need to say a massive thank you to a very fantastic group of people that have been working since March and are behind a lot of the work that I'm going to talk about. So there's Nick and Andrew, Christoph, Alex, and we also work closely with Achilles Kapanidis, and he runs the gene machine groups in biophysics. So without all of these people, I really wouldn't be here to kind of tell you the story. So thank you very much to them. So I think it's safe to say that at the moment, viruses are probably on most people's minds. Wherever you are, whatever you're doing, you definitely would have been affected by the current coronavirus pandemic. But viruses have actually been around for a long time, and they've always represented a huge global health concern. So there are other viruses that there's always been a constant threat of epidemics or pandemics, and some of the ones that have really hit the headlines over the last couple of years are influenza. There's the Zika virus, which has been associated with sort of neural defects in babies born to mothers who are infected with the virus. There's Ebola, which has caused a huge outbreak in Western Africa since 2015, and that's still ongoing. And there's another coronavirus which causes something called Middle Eastern respiratory syndrome, which is sort of centered more around the Arabic Peninsula. But I think that none of these have really had as much of an impact as the current coronavirus or COVID-19 pandemic, which is really the defining global health crisis of our time. So it's thought that it's emerged in the Chinese city of Wuhan late last year, and since then it's spread to every continent of the world except for Antarctica. So when I was putting these slides together a couple of days ago, at the time we'd just passed 7 million confirmed cases. This has now gone up when I looked today, and we are well over 400,000 confirmed deaths. So this is actually a huge underestimate because of sort of large differences in testing capabilities in different regions. But just important to point out that we're definitely on an upward trend, and we haven't shown any sign of reaching the peak of number of cases yet. So I'm going to just talk a couple of slides about the virus that causes this COVID-19 pandemic. So COVID-19 is a respiratory illness. It's spread between people primarily through aerosols, which are produced when you cough or sneeze or talk to someone. So this really quite disgusting picture of a sneeze really highlights why you need to cover your mouth if you're coughing or sneezing. The symptoms for most people are quite mild. They include fever, fatigue, a cough, loss of taste, for example. But in a very small number of people, this can progress into a much more serious illness called acute respiratory distress syndrome, and that can be fatal in a small number of individuals. So the COVID-19 is caused by a very small virus with a very, very long, complicated name. So it's the severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2 for short. And it's an envelope virus. So viruses are very tiny parasites that can't reproduce or survive for long outside of a host cell. And you can essentially think of them as just a ball of protein. So in this case, the SARS-CoV-2, the ball of protein is also surrounded by an envelope of lipids, which is shown in grey in this image. And sticking out of the envelope are these red protrusions, which are the spike protein of the virus, and that's important for the next slide. And inside that ball of protein is the genome of the virus, which in this case is made up of RNA. So one of the important questions that's come out of this pandemic is whether infection with SARS-CoV-2 actually makes you immune and can stop you getting reinfected with the virus a second time round. So those red protrusions, the spike proteins, are the primary target for antibodies that can give you immunity to the virus. So there have been some studies in monkeys which suggest that once you've had the illness and you've recovered and then you get re-challenged with it, those monkeys didn't get ill again. So there is some anecdotal evidence that immunity does exist, at least in the short term, but it's not really clear with this applies to humans or how long that immunity might last for. So what Andrew and the group has been doing is some modeling. So he's extended the generic SIR framework, which is the susceptible, infected, and recovered, to also include people that are infected 2 or more times and then recovered 2 or more times. And he's used this to investigate the reinfection frequency of the disease. And when he did this for different regions around the world and different countries around the world, what we found is that the model predicted that a small number of reinfections should have occurred by now. So the important point is that until now, no cases of reinfections have been documented anywhere. So this actually suggests that exposure to the virus does actually provide at least a short-term immunity. We can't say anything about long-term immunity because not enough time has gone by. So now I'm going to switch from this to talking a little bit more about the experimental side that we do in the lab. So like I said, viruses are extremely tiny. So SARS-CoV-2 is on average about 125 nanometers in diameter. And this is so small you can actually fit many millions of them onto a single pinhead. It's also smaller than the diffraction limit of visible light, which is something that I'll come back to. It's important for imaging. So because they're so tiny, the techniques that we use are single molecule techniques. And these are really ideal for looking at viruses. You don't get any averaging. You can capture really rare and transient events. You don't need to synchronize your population. And of course, with advancements in microscopy, we have really the ultimate sensitivity of looking at single particles without any amplification. And we combine these methods with fluorescence, which is just a type of luminescence where emission wavelength is longer than excitation from the excited light-induced singlet state. And together, this gives us a toolbox for studying viruses. So I think the current pandemic really highlights how important it is to have easy, sensitive, rapid ways of detecting viruses. So I'm just going to talk a little bit about the methods that are currently available. So one way that you can see if you've got a virus there is to directly look for it by using electron microscopy. You can grow up the virus in virus culture in cell lines. For some viruses, so for influenza, for example, there are rapid tests that look for viral antigens using antibodies. I must say that there are none of these available for SARS-CoV-2 yet. And finally, you can detect the genome of the virus. So this is done through a technique called RT-PCR, where you amplify up the genome and use fluorescent techniques to detect it. And this is currently the gold standard for SARS detection. So this is what is used in laboratories worldwide to tell if the virus is present in a sample or not. There's a lot of pros and cons to all of these methods, but what they have in common is that they take quite a significant time to give you a result. So things like growing the virus can actually take days, if not weeks. Microscopy can take hours, and even the so-called rapid tests take at least 30 minutes to give a result. And those are hampered by having quite low sensitivities and specificities. So what we aim to do is use our single molecule fluorescence techniques to detect intact virus particles. So they don't need to be grown up, they don't need to be broken open or purified, for example. We just see, we have the sensitivity to see the intact virus particles in a sample straight away. So the basis of our first assay is that viruses, although they are all very small, they actually come in quite a range of different sizes. So this can range from a tiny parvovirus, which is only 20 nanometers in size, all the way up to Ebola, which is actually almost a micron. And the virus that we've used in this initial test is influenza, which is around 120 nanometers in size. So the concept of the assay is that we fluorescently label the exterior of virus particles, and we use single particle tracking to watch the viruses as they diffuse randomly in solution on our microscope. The single particle tracking, you localize the visible spots in each frame of your acquisition movie, and then you associate the spots with spots in adjacent frames, and you get trajectories. And from this, you can get a diffusion coefficient for each trajectory. We then plot these. We take the peak as the average diffusion coefficient, and we use the Stokes-Einstein relation to get an estimate of the diameter of the diffusing particle. And that's the readout for our assay. So for this to work, what we need is a really quick and easy way of getting bright fluorescent virus particles. So this work was done by a former MPhil student in the group, Jonathan Taylor. And what Johnny was able to show was that if we take an envelope virus like influenza and we add a positively charged cationic solution like calcium chloride to it, as well as short DNAs, and those DNAs are modified to contain a fluorophore, the calcium chloride will actually bind those DNAs to the surface of the virus, and you get a fluorescently labeled particle. So when we do this, and when we look at, this is diffusing flu viruses on our microscope, you can see that we have beautiful single particles diffusing around. Any unbound DNA that contains a fluorophore that's not bound to a particle will basically diffuse much, much faster, and so it just blurs out. You can't track it. However, the labeled viruses, which are labeled with multiple DNAs, so they're very bright, and they're obviously much bigger than free DNA, diffuse more slowly, and these are easy to track. And the really excellent thing about this labeling is that it's instantaneous. So as soon as you add your virus to the sample, they're instantly labeled and you can observe them on a microscope. So this is much, much quicker timescale than any of the other existing methods out there. The labeling is also specific to viruses. So if you leave a virus out of the solution, or you leave out the calcium chloride, or you leave out the DNA, you don't get any signal at all. And you can also reverse the reaction by adding a chemical called EDTA, which chelates the calcium ions out of the solution, and then you lose all of your labeled virus particles. When we tracked flu diffusing in solution, we got a Stokes diameter of around 134 nanometers. And when we went and looked at the virus particles using electron microscopy and measured the average diameter, we were also able to show that the results we got basically corresponded to particles of the correct size. So this was a really nice, quick, easy way of labeling viruses. Put them on your microscope and you get a readout, and that tells you the estimated size of the virus that you're looking at. We wanted to understand a little bit more about why the labeling worked. So we did some experiments where we replaced the calcium chloride with other cationic solutions, for example, sodium chloride or magnesium chloride. And what we showed is that really we only got efficient labeling when calcium chloride was present. We also tried to replace the DNAs with either RNA or fluorescently labeled protein or just free fluorescent dye. And again, we saw that we got some labeling with an RNA present, but mostly the labeling worked best with DNA. So it seems to work with nucleic acids in general, although sort of favored towards DNA. And finally, we investigated the need to have the viral envelope. So some viruses don't actually have the lipid envelope. For example, adenoviruses don't. When we tried the labeling with that, we couldn't see any labeled particles. And we also used small artificial vesicles that we produced in the lab to have a certain charge. So anionic vesicles with an overall negative charge labeled beautifully, but if we had vesicles with either a positive charge or a neutral charge, these didn't label at all. So taken together, it looks like we need a cationic solution, a divalent cationic solution like calcium chloride, DNA, and an envelope to get labeling. So we've come up with this scheme for how we think the labeling works, where basically the calcium ions from calcium chloride solution, which are positively charged, mediate an interaction between the negatively charged phosphate backbone of the DNA, as well as the negatively charged phospholipids in the envelope. To investigate our limit of detection, we looked at increasing concentrations of virus, and what we saw is that when you leave out the virus, you get absolutely zero tracks. So this gives us our limit of blank, it's called, and this can then be used to calculate the limit of detection, which is basically the smallest number of particles that you can reliably detect above background. And for this, we get a limit of detection of around 10 to the 4 particles per mil, at least in our tracking assay. And we moved on to look at clinical samples of influenza. So these are throat swabs that were provided by our collaborators in Greece. And for this labeling, because the concentration of virus in these samples was a lot lower than the samples that we'd been using previously, which was laboratory grown strains, we actually used 2 colors to label the virus. So we used 2 DNAs simultaneously, some of which were labeled with a green DNA and some of which with a green dye, and some of which were labeled with a red dye, just to make sure that virus particles could be detected reliably by having both colors on them. So you can see in this image that we get nice co-localized colors in red and green when virus is present, but you don't get anything when the virus is left out. This is just showing the co-localized tracks. And what we're able to show is that the assay worked really nicely to detect flu in clinical samples, and overall we could detect, reliably label and detect a virus in about 1 minute. So this is a significant improvement on any of the other viral detection methods that are out there so far. This is also a really nice way to study virus aggregation. So different viruses aggregate to different extents. They're just generally sticky, little sticky blobs. And when we looked at different strains of influenza, we saw that the Stokes diameters we were getting out were much bigger than we expected for single particles. And also by eye, you could see that some of the the spots that we were tracking were bigger than single particles. So we hypothesized that we were actually looking at sort of mixed population of some single particles, but also small aggregates of viruses as well. That's just highlighting the size of the particles that we're seeing, which is a lot bigger than the 120 nanometers or so we're expecting. So this was supported by some simulations that Amy, another info student in the group, did, where she simulated the movements to mixed populations of labeled particles, and she was able to show that the results we were getting were from a mixed population of small aggregates rather than just single 100 nanometer diameter particles. Because our fluorescent labels are so bright, and also because we're getting multiple labels per particle, in fact, our viruses are as bright as sort of microsphere beads that we actually use to calibrate our microscopes, which are pretty much the brightest thing that we can get hold of. And we did a quick kind of back of the envelope kind of test to try and determine how many DNAs we were getting per particle by looking at the size, the intensity profiles of immobilized viruses versus just immobilized fluorophores, and just integrating under the area of each particle. And we're able to show that for DNA only, so essentially fluorophores only, we got a certain intensity, and this is much higher when we looked at virus particles, and we're able to estimate that we have around 30 DNAs per virus. This is really useful for something else which you can do, which is virus counting. So rather than looking at just all the particles diffusing around in a field of view, we built a kind of simple fluidic system where we use a syringe pump to pump the viruses through a narrow microfluidic channel, and we use a confocal microscope which alternated red and green laser excitation to look at each virus as it flowed past. So every time a virus passes through the confocal volume, we get a burst of fluorescence in both the red and the green channel because the viruses were dual labeled. And we can use this as a really neat way to quickly quantify how many viruses you have in a sample. So as well as looking at diffusing viruses, we have another technique, which is to look at viruses that are immobilized on the surface of a slide. And we do this using something called turf microscopy, or total internal reflection fluorescence microscopy. So here the incident light beam is shone at the sample at an angle greater than the critical angle, so that the light is totally internally reflected. However, you get a exponentially decaying evanescent wave at the same frequency, which passes into your sample. And because this is decaying, it only passes into the sample within about 100 nanometers of the surface. So you can see in this image here, so you have your sample on the microscope objective on a glass slide, and this is a really, really neat way of making sure that you only see whatever is stuck to your glass slide and you minimize any background above it. So we can do this with viruses. However, because viruses are so tiny, the images we get are diffraction limited. So when light from any point source is shone through an aperture like a lens on a microscope, it's blurred out to a particular 3D shape known as the PSF, point spread function. And in a set of physical laws defined by Ernst A back in 1873, he showed that the diffraction limit was around 250 nanometers for visible light, which is basically the full width half maximum of the PSF. And for two objects to be resolved, the Rayleigh criterion basically states that the two PSFs need to be further apart than the width of one of the PSFs. So this is a bit of a problem when we're looking at such tiny things like viruses. But there are some microscopy methods that are designed to get around this. And together these are called super resolution microscopy. So one of the examples that I'll show you is a technique called STORM, and this is something that we use quite commonly in the lab. So if you have a image where all the fluorophores are switched on at the same time in your sample, they're going to be overlapping and they're going to be so close together you can't resolve any individual fluorophores. But if you use dyes that blink, what you can do in each frame of your acquisition, only a small subset of the dyes will be switched on. So because these are far enough apart, you can precisely localize using the centroid of the localization exactly where they are. And then after that, this small subset of fluorophores that switch on will then be deactivated and switched off. And you can just cycle these. So in the next frame, another subset of fluorophores are switched on. If you do this, you can build up a reconstructed super resolution image where you add up all of your very precise localizations in your sample, and you can get resolutions of lower than 20 nanometers. So this is just one example. These are calcium and DNA labeled virus particles stuck on the surface of a glass slide. That's the diffraction limited image that you get. And this is the super resolution image of the same field of view. And when we zoom in, we see something really interesting. So what looks like just a single virus particle to us is that it's actually labeled in a way so that the labeling isn't universally covering the whole surface of the virus particle. So we take advantage of this in the next assay that I'm going to talk about. So the next thing that I'm going to describe is based on work done by primarily by Nick in the lab. So he joined as an info student. He stayed on to do a DPhil with us. He's had a helping hand from Alex, who's an info student that we've had in the last year. So Nick's idea was really to see if we can develop a faster way for detecting viruses using a number of things. So the first was to use diffraction limited virus images. So we don't need to use any fancy techniques like super resolution, which take time. We're just going to use the raw images, which are diffraction limited. We're going to take multiple images really quickly in an automated fashion using multiple acquisition capability of our microscope. We're going to have batch analysis methods that are, again, automated and extremely fast. And finally, we're going to use deep learning to classify the images of virus particles that we see. And this is the basis of the assay that we've been developing over the last couple of months to detect coronaviruses. So the schematic at the bottom shows how our labeling and sticking of viruses works. So we label viruses using the calcium chloride and DNA method that I described previously. These are stuck down onto the surface of a glass slide that's been treated with a polymer called chitosan that basically makes the glass very sticky and it sticks the viruses down very quickly. And then we use a turf microscope to look at them. Oh, just to mention also, we use, again, the dual color labeling. So we use a mixture of DNAs that are labeled with both a red fluorophore and a green fluorophore to make sure that virus particles are labeled with both colors. And then we can distinguish them from just free DNA, which would only be a single color. So we started off a couple of months ago working on coronaviruses, but we didn't want to sort of start out straight away working on the real thing. So we've got a kind of safer alternative coronavirus, which is called IBV. And this is an avian coronavirus that doesn't infect humans, so it's quite safe for us to work with in the lab. These are diffraction limited images of IBV. So they're labeled with red and green DNAs, so you can see signals in both the red channel and green channel of the microscope. And then in the third panel down is the merged localizations from both channels. So you can see single reds, single greens, which correspond to just free DNAs, and also yellow, which is the merged localizations where you have co-localization between red and green, and this corresponds to labeled virus particles. If you leave the virus solution out, you again just get single reds and single greens, but you don't see these co-localized signals. Similarly, if you leave out calcium chloride, you just get red and green, and if you leave out the DNAs, you don't see anything at all. So apologies for my very wobbly video here, but this is just to demonstrate the multiple acquisition capability of our microscope. So basically, we prepare a sample and then to remove any bias in our sampling, and also to make sure that we can take a lot of images very quickly, we use multiple acquisition, which where the microscope automatically changes the field of view, refocuses, and takes a short movie. And using this, we can take around 81 fields of view of a single sample in around 2 minutes. Following this, we have batch analysis methods to segment our images and get them ready for further processing. So we start off with raw fields of view, as shown here. So this is just the raw diffraction limited image. We then use intensity filtering to binarize the image, and area filtering to exclude objects with of a pixel size below 10 or above 100. So this effectively gets rid of any single DNAs, which are very tiny, or any very large aggregates, which might be large aggregates of virus, but might also just be other kind of cellular junk that we want to discard. We also get the location image corresponding to the raw image, which shows the single green, the single red, and the co-localized signals, and from this we can just use the co-localized signals. We use segmentation to isolate the signals into bounding boxes, or BBXs for short, and in this way we can discard any signals coming from just single red or green, and just use the co-localized signals going forward. So this is just an example of the co-localized bounding boxes that we then use for our deep learning. See if we can go to the next slide. Oh, also just to mention that this is extremely fast, so each field of view that we take is processed in around 2 seconds. And this is just a quick plot showing that when we compare the labeled IBV virus with our controls, where we leave out virus or calcium chloride or DNA, we get a much higher number of co-localized signal when virus is present. So because the sort of early symptoms of COVID-19 are quite generic, shall we say, so that they can really be attributed to many different viruses. It's really important that in our tests we're able to distinguish between coronavirus and other viruses as well, and particularly if it's useful to be able to distinguish between coronavirus and flu, which has very similar early symptoms. So what we started with doing was using laboratory grown viruses. So we have our safe alternative IBV coronavirus and 3 strains of influenza, and we imaged them, took multiple images, segmented them, and then looked at the properties of the bounding boxes that we got. So we looked at maximum pixel intensity, the area, and the axial ratio, and we could actually see that there were very small subtle differences between them. So the idea was that we would take advantage of these small differences, as well as any other differences that you can't actually see by naked eye, and use machine learning to distinguish between the images and hopefully be able to classify viruses in this way. So we built a shallow convolutional neural network. It's a 15-layer network, and this is just a schematic of what it looks like. So there's an input layer, which is basically the bounding boxes from the segmentation process. There are 3 convolutional and ReLU layers, which is to introduce non-linearity into it. The first 2 are followed by a max pooling layer. And then we have a fully connected layer, a softmax layer, and a final classification layer to decide what it is that we've got. This is just an example of the validation. So for our 4 virus strains that we're trying to distinguish between, so 1 coronavirus and 3 flu viruses, we started around 25%, which is what you'd expect with a random classification of 4 different objects. You get an initial increase as the network sort of learns the easier kind of parameters, and then this slows down over sort of subsequent iterations. At the same time, the loss function gets minimized. And yeah, in this case, it took about an hour and a half to train the network. I think it normally takes around about an hour. So we show our results as confusion matrices. So that's described here. So we plot the output class, which is the predictor class, against the target or the true class. And we distinguish between our true positives, bounding boxes that are true positives, true negatives, as well as false positives and false negatives. And we also have a positive and negative predictive values, which tells us how well our network is working, as well as a measure of the sensitivity and specificity, where sensitivity is essentially the number of positive cases who actually test positive, and specificity is negatives who test negative. And then in the bottom corner there, we have the overall validation accuracy, which is the balance accuracy of the results. Also, just to point out that the overall accuracy that we get is the probability referring to single bounding boxes or single virus particles in the sample rather than to the whole sample itself. So if we started with our laboratory grown viral strains, because they're the easiest to work with. Again, just to remind you, we've got coronavirus and 3 flu strains. We showed that we could really nicely distinguish between IBV and the negative with around 92% validation accuracy. So that was really exciting. And similarly, if we pooled together the negatives with all of the flu strains, we could really nicely distinguish IBV from that as well. So that's quite exciting from a kind of clinical point of view. So we'd be able to train a network to pick out coronavirus and distinguish it from other types of viruses that might be present in a sample. When we compared IBV with one of the flu strains, which is called Udorn, we could very nicely again distinguish between the 2. And when we compared between flu strains, so between different subtypes of influenza, we had slightly lower validation accuracy, although overall the network was still able to distinguish between them to about 70%. So we started off in the lab working with our laboratory grown strains. We showed that the assay worked, and then the next thing was that we wanted to test actual real life COVID-19 samples. And so for this, we had a lot of help. We've got a lot of people really to thank for this. So we had the loan of a microscope from the Micron facility in the biochemistry department, and we moved that to the John Radcliffe Hospital, and we set it up in Derek Crook's lab. So he is the head of the research microbiology lab at JR, and he very kindly hosted us there. And so that's where we've been working for the last couple of months. So moving from laboratory grown samples to clinical samples definitely provided some challenges. So I'll just go over a couple of them quickly now. So for a start, working with our laboratory adapted and the laboratory grown strains is much safer than working with real SARS-CoV-2 samples. So the samples needed to be inactivated via a validated protocol. So different laboratories and different pathogens are divided into different biosafety level groups, with biosafety level 1 being for non-pathogenic organisms, and biosafety level 4 being for very exotic, dangerous pathogens for which there's absolutely no cure or no treatment. SARS-CoV-2 is classified as a BSL-3 pathogen. So this means it needs to be worked with in a high containment lab, and our microscope was in a BSL-2 lab. So one of the first things that we needed to do was sort out how we could actually get the samples from the BSL-3 lab to the BSL-2 lab to work with them safely. We also noticed that there was a lot of heterogeneity in the clinical samples. So different samples that were coming into the hospital lab were from different types. So you have throat swabs, you've got nose swabs, and you've got something called a nasopharyngeal aspirate, which is basically a vacuum pump of your nasal cavity. And what this meant for us is that, and for our machine learning, is that different samples have a lot of different backgrounds in them. They're also, because they're clinical patient samples, there's the fact that other viruses might be present in there. So some examples of flu, RSV, other low pathogenic coronaviruses that cause the common cold would be there as well. And also the clinical samples have a much lower concentration of virus particles in them than our normal laboratory grown strains that we're used to working with. And so the first thing that we had to do was sort out the virus inactivation. So as I mentioned, you have different types of samples coming into the lab, and they all go into the BSL-3 lab. And from there, most samples are inactivated. The virus is essentially lysed and burst open so that the genome can be accessed for RT-PCR. However, for our assay, this is no good because we need to have the intact virus particles to label and image. So we came up with a new inactivation protocol, which was to fix the intact virus particles in a chemical called formaldehyde. And this then rendered them fully non-infectious but still intact, and we could then move them into BSL-2 for imaging on our microscope. And we're able to validate that formaldehyde treatment actually did inactivate the virus. And this was work done by some collaborators of ours over in France at the University of Montpellier. So they used infectivity assays to show that the treated virus couldn't go on and infect cells. So having overcome some of these initial challenges, Nick started working his way through the clinical samples. And what he could show is that we can use a trained network to differentiate between SARS-CoV-2 positive samples and negative patient samples with an overall validation accuracy of around 70%. We get a similar result when we look at patient samples that have other coronaviruses in them. So these are human coronaviruses that aren't as serious as SARS-CoV-2. So they cause, they're one of the leading causes of the common cold, and there's different strains of these, which are just listed there. So when we differentiate between these kind of common coronaviruses and negative samples, we can distinguish between them with around 77% validation accuracy. So overall, we get a lower validation accuracy than we do for laboratory grown viruses. And this really reflects some of the things that I mentioned before, the heterogeneity in the samples, the lower concentration, for example. And one of the kind of exciting things that we could do is we can also distinguish quite reliably between SARS-CoV-2 virus and the other common human coronaviruses, which again is really important from a sort of immunity point of view, so if there's cross immunity between different strains, and also from a testing point of view, so you can tell exactly which virus a patient is ill with. So finally, this is just something that we've literally been working on in the last couple of days, so it's just quite preliminary, but having trained and validated our set on our initial clinical samples, we then wanted to carry out a blind test to see how well our network did with data that had never seen before. And so we captured images of the viruses on 4 different days. So we have samples from patients that were ill with SARS-CoV-2, with other coronaviruses, as well as negative samples that had no virus in them. We used the data from days 1 to 3 to train the network using around 20,000 bounding boxes per category. And we divided that data into 70% used for training and 30% for validation. And then we used a random sample from day 4. So this is data that the network has never seen before during the training or validation to do a blind test. And we were able to show that samples that were positive were predicted as positive, and those that were negative were predicted as negative. So we need to, this is very preliminary, like I said, we need to repeat this on a lot more samples, but that's pretty exciting. So this is the basis of the clinical test that we're trying to develop. Right, so that just leaves me to summarize. So we have a method using calcium chloride to very, very quickly fluorescently label virus particles. We can use this method to track virus particles in a tracking assay where we get a readout in less than a minute. We can also use automated imaging and batch analysis methods on immobilized and fluorescently labeled virus particles, and use machine learning algorithms to classify viruses where we get over 90% validation accuracy and very high sensitivities and specificities for our lab-grown strains, slightly lower for the clinical samples that still needs a bit more optimization. And overall, the timescales of our tests are significantly shorter than any existing tests out there. So they can just be completed in just a matter of minutes compared to hours or days or even weeks. This is just a couple of fun photos of Nick and Andrew and I at the hospital taking some data. And finally, I have a lot of people that I need to thank. So members of my group, Andrew, Nick, Alex, and former members Amy and Johnny, members of Achilles group, all the clinicians at the John Radcliffe for letting us use their lab space and providing us with samples, the Micron facility in biochemistry for the loan of their microscope, and a lot of external collaborators as well. So Erica from Pirbright, who provided us with the IBV virus, our collaborators in pathology that gave us some cultured CoV-2 virus, collaborators at the University of Montpellier that helped us with the virus inactivation procedure, and other Greek collaborators that helped us with clinical samples of influenza. And finally, our funding sources, which comes from the Royal Society, as well as some funds from the Oxford University COVID-19 Research Response Fund. And thank you all for listening.